Now, when you look at bone under the microscope, bone tissue, you're going to be looking at both compact bone and spongy bone. Spongy bone is going to look a lot like hyaline cartilage, except that you'll notice that it's in these kind of thin branching lines, almost like a spider type of a network. If you take a look at the trabeculae of spongy bone, right, no other cartilage ever looks like this. So when you see these thin lines of collagen, osteocytes living in lacunae, again, the bone itself will kind of have a similar appearance to cartilage when it's stained, especially with H&E, with the pink collagen in the extracellular matrix, the osteocytes living in lacunae. But that is going to be spongy bone when you see these thin little, almost like spider web network. Compact bone has its own unique characteristics. One of the things about compact bone that you'll notice is that we rarely need to stain it because it has such a defining characteristic. When we slice across compact bone in a cross section or a transverse section, you'll notice that compact bone looks a whole lot like a bunch of tree trunks. You know, just like the rings of a tree, these rings of compact bone are arranged in a very similar pattern, and that's what gives compact bone its incredible amount of strength. Each one of these sets of rings, like a single tree trunk. Let me get my pen. So each set of rings here is called an osteon. And you can see that an osteon is three-dimensional, right? It's more of a cylinder. But if I look at a cross-section of an osteon, it's going to have this appearance here, or under the microscope that we have, our light microscopes, it looks like this image on the bottom here. We're going to find a central canal in the middle. That central canal is a hollow space that contains arteries, veins, and nerves. These central canals of osteons provide the passageways for arteries, veins, and nerves in our bone tissue, in our compact bone tissue. Surrounding the central canal are going to be rings of extracellular matrix. The ring of extracellular matrix is called a lamella, or sometimes they're referred to as the lamellar rings. So you see this ring-like pattern. There may be 3, 4, 5, right? A handful of rings surrounding a single central canal. The rings, if you follow the lamellae, lamellae is plural, drop the E, and it becomes lamella to say one ring. If you look around each lamella, you will notice a little bubble, a little lake. We call that a lacuna. And inside of every single lacuna is going to be one osteocyte. The osteocytes live in lacuna, and the lacuna are arranged around each lamella. So if you take a look at the light microscope image on the bottom, every single black spot is an osteocyte living in a lacuna, and each lacuna is going to circle the lamellar rings. We have one more thing to mention here, and that is a problem, really. How does nutrients, how does oxygen and carbon dioxide, nutrient molecules, waste molecules, how do these things diffuse through this mineral? The ground substance of bone has no water, right? This is not a firm gel. This is mineral. So how do we get the nutrients and materials that this osteocyte living in this lacuna way out here, how do we get that material to this guy? It can't diffuse through mineral. It can't diffuse through the calcium crystals. What happens is these cells touch one another. Look at the osteocyte in the illustration up above. It's got these little kind of foot-like extensions that branch away from it and basically connect to neighboring osteocytes living in lacunae through little tunnels or little canals that are kind of tunneled through the lamellar rings. The little canal is called a canaliculi. Osteocytes touch one another via canaliculi. when we look at the bottom image, all these little black lines that you're seeing, they kind of look like spider legs. Those are the canaliculi. So nutrients can travel through this osteocyte and through the canaliculi to the next lamella level of osteocytes, and then through the canaliculi to the next lamella level of osteocytes, and so on. And this is actually what limits the number of rings. You're never going to see a lamella, or I should say an osteon, that's this big. They don't exist. And that's because all of the nutrients from the central canal's artery would be lost. They would be sucked up and drained by the time they reached the outer layer of the lamella. So again, you limit the size of your osteons by allowing this nutrient to pass from one cell to the next to the next. Every cell along the way takes what it needs and then passes on the leftovers to the next cell. And so that, again, eventually you're going to run out of nutrients. The lamellar rings can only be so many, can only be so big. Each osteon can only be so big. I can't really illustrate this in a cross section, but this image up here also shows a longitudinal section through compact bone, the side view here. And in the side longitudinal view, you can see what's known as perforating canals. So the central canal is in the center of each osteon. The perforating canals are lateral to those central canals, and that's what allows arteries, veins, and nerves to connect up from one central canal to the next to the next. So we get these pathways through the bone's diaphysis in this way, through those perforating canals. We talked a little bit about red and yellow bone marrow already. Remember that yellow bone marrow is going to fill the inside of our medullary cavities, and red bone marrow is going to fill the spaces within the spongy bone at each epiphysis of our long bones. And I already mentioned that we have these medullary cavities primarily to lighten our bones. If our bones were densely packed all the way through with compact bone, they would be much, much heavier, and our muscles would have to be really big to be able to move the heavy bones of our bodies. So it lightens the load to have your bone be hollow. The cells of bone that we've talked about so far have been osteocytes. Osteocytes are the mature cells of bone. They live in lacunae, and remember that they touch one another via those little canaliculi, through those little tunnels. Osteocytes arise from an immature cell known as an osteoblast. Osteoblasts are generally going to be found located in the endosteum layer of dense irregular connective tissue lining the inside of our bony surfaces, and the periosteum. The periosteum is the outer layer covering the outside of our bony surfaces. So osteoblasts are going to be found in the periosteum outside of our bones and in the endosteum inside of our bones. Bone growth can occur through these osteoblasts. When an osteoblast is stimulated to begin to deposit and build new bone, it does so by building bone from its surface, from the endosteum surface and from the periosteum surface. So bone will continue to get thicker and thicker and thicker as the osteoblasts lay down new bony matrix. So think of B, the blast, as the bone builders. Now, osteoclasts are a different type of bone cell. They are not at all related to osteoblasts and osteocytes. Osteoblasts will actually mature and become an osteocyte as soon as it begins to lay bony matrix and traps itself in a lacuna. Osteoclasts are a completely different cell type altogether. They're actually derived from a cell of your immune system called a monocyte. The osteoclasts, these guys live in endosteum and periosteum as well. So they're found in the same coverings, the dense irregular connective tissue. But their job is to digest bone. When they digest bone using hydrochloric acid, they're going to be able to break up the matrix. So this is an osteoclast, this big purple cell, and it's actually digesting the matrix by secreting hydrochloric acid. When it digests the matrix, it allows bone to release calcium and release phosphates into the bloodstream, into circulation. So osteoclasts are really, really important because if our blood calcium levels get too low, then our neurons are going to stop functioning and our muscles are going to stop contracting. Both of those really important cells for your survival require calcium for their function. So we have these huge stores of calcium throughout our bodies. This is how we tap into those calcium stores. Osteoclasts are the bone chewers. So C for chewing, blast, the B is for building. And there's a constant back and forth between these two, again, depending on blood calcium levels. When blood calcium levels are high, osteoblasts store that excess calcium by building bone, and our bones get thicker and stronger. But when our blood calcium levels get low, osteoclasts can digest that exact same bony matrix to be able to release the needed calcium to support the continued functioning of these really important cells for our survival, our neurons and our muscle cells in our bodies. How does bone form? So the formation of bone is called ossification, and there's two different processes. This all occurs before we're born. There's intramembranous ossification that occurs for the flat bones of the skull, and your clavicles develop in this way. All the other bones of the body are going to develop from something called endochondral ossification. Endo is within, chond, right? There's our big root word here for cartilage. So endochondral ossification is bone formation from within cartilage. Remember that I mentioned at the beginning of this lecture that almost all of the fetal skeleton before we're born is laid out as hyaline cartilage models. Every single bone in the body with a couple of exceptions. Well, these are the exceptions, the flat bones of the skull and the clavicle. All the other bones of the body are laid out in hyaline cartilage, and that cartilage begins to ossify. So that's going to happen before we're born, and then will continue to happen all the way through our childhood, actually, as our bones continue to elongate and grow. So let's start with intramembranous. Intramembranous ossification occurs from within this membrane-like tissue called a mesenchyme in the embryo. In the very, very early embryo, there is a connective tissue proper called mesenchyme. It's an embryonic connective tissue, and it's got a lot of stem cells. Well, when some of those stem cells are stimulated to become osteoblasts, that's what's going to stimulate intramembranous ossification. Some of the stem cells within the mesenchyme are going to be stimulated to become osteoblasts. The osteoblasts, as a cluster, form something called an ossification center. Remember, osteoblast's job is to build bone. So they start to lay down the calcium matrix, they lay down the collagen matrix. They're also dividing through mitosis, so you can see that their numbers increase. Well, eventually, the osteoblasts will find themselves trapped in the matrix that they're building. When they do, they mature into an osteocyte. So we've got osteoblasts becoming osteocytes, more osteoblasts continuing to lay down more matrix, continuing to divide and increase in number. And this is when our bone begins to calcify. We'll start to see blood vessels get drawn into this calcified developing bone, still happening in the embryo, right, in the very early developmental stages in utero before we're born. This blood vessel invasion is what's going to carve out the spaces. Bone will form around all of the blood vessels, and that's what leads to the spongy bone appearance of woven bone or spongy bone. Now the ossification will continue, and we're going to end up seeing a little layer of compact bone on either side. Our osteoblasts, which have been continuing to grow in number, are going to remain in the periosteum so that this flat bone can continue to get thicker and thicker and thicker through these osteoblasts that are living out here in the periosteum layer. Again, bone has formed on the inside, and we've got this spongy bone surrounded by compact bone on either surface. That's how the flat bones of the skull form in this in this intramembranous ossification way. Here's an example of a flat bone of the skull. You can see that the superficial surface of the bone is going to be compact bone, and the deeper surface of the bone is going to be compact bone. Periosteum is still going to be covering the bone on all surfaces, but the inside of the bone has this spongy network. The spongy bone of the flat bones of your skull is sometimes referred to as the diploe. It does contain bone marrow in all of those little cavity spaces. We have red bone marrow in the diploe, but there's no marrow cavity. There's no yellow bone marrow in these flat bones of the skull. Other irregular bones of the body have a very similar appearance with spongy bone on the inside and a thin layer of compact bone on the surface. Irregular bones are these bones that have irregular shapes, like your vertebrae, for example, would be a good example of an irregular bone. The other form of ossification is endochondral, from within cartilage. I mentioned most of the other bones of the body are going to be laid out as a hyaline cartilage model, essentially, or precursor. So a long bone might be a hyaline cartilage model in the early embryo. What happens to that fetal precursor is ossification. So bone will slowly start to take over the cartilage. One of the things that happens is as the cartilage grows and grows, it begins to deteriorate and calcify. And that is going to happen primarily, well, it will happen first when a bone collar forms in the diaphysis region of this long bone. The bone collar begins to slowly kind of replace cartilage with bone tissue in the diaphysis. The invasion of blood vessels is what ends up carving out the medullary cavity eventually in the diaphysis of our long bones. As soon as those blood vessels begin to invade our long bone, we're going to see a secondary ossification center beginning to form in each epiphysis end. So the epiphysis will then ossify. And again, the blood vessels in the epiphysis are what are going to carve out all of the cavities of spongy bone in the proximal epiphysis and in the distal epiphysis. So we've got 3 ossification centers in our long bones, 1 primary ossification center and 2 secondary ossification centers. Between the epiphysis and the diaphysis is still some leftover hyaline cartilage. This is the same cartilage that was originally there in the hyaline cartilage precursor. Our bones look like this when we're born. Bone tissue in the epiphysis, bone tissue in the diaphysis, but hyaline cartilage in the metaphysis area. This is referred to as the epiphyseal plate of hyaline cartilage. And that epiphyseal plate will not fully ossify until we've reached our final height. Usually, we stop growing, we stop elongating right around our early adolescence stages. So late teens, early 20s, that's typically when the epiphyseal plate fully ossifies and we have this epiphyseal line as mature adults. Again, in both epiphysis ends, or both epiphyseal ends, proximal epiphysis and distal epiphysis. So this collectively is known as endochondral ossification. It is the formation of bone from within cartilage. This epiphyseal growth that's going to occur from the time we're born until we stop growing, again in early adolescence, is going to happen in our long bones at both ends. The way that that works, if we looked carefully at an epiphyseal plate, you would find, let's say that this is a proximal epiphysis. You would find the proximal epiphysis up here on this surface, and we've got this zone of hyaline cartilage adjacent to the proximal epiphysis. So just for reference, we would be looking at this region right here in this particular bone, if this were taken from that bone. We've got a zone of resting cartilage. Not much is happening here, but you'll notice that adjacent to that zone, closer to the diaphysis, The chondrocytes are dividing like crazy. This is our proliferating cartilage here. And when chondrocytes divide, remember they live in lacunae, they don't like to be next to each other. So they very quickly begin to lay down more and more matrix, and they start to spread away from each other in this proximal distal pattern. So we end up getting these basically rows or stacks of chondrocytes. Then, as the chondrocytes are stacked up like this, they begin to die. They get close to the bone tissue down here, and they begin to die. When they start to die, the first thing that a dying cell often does in our bodies is it puffs up. So hypertrophy means to grow or enlarge. We'll see that the dying chondrocytes get puffy. They get a little bit larger first, and then they actually do die. When they die, it's because they've calcified, or the matrix around them has calcified. If there's no more gel ground substance, then there's no ability of materials to move and diffuse to keep these chondrocytes alive. So the calcification of the ground substance is what leads to chondrocyte death. That calcification, so we have calcified cartilage here, will eventually be completely taken over by the osteoblasts that are going to continue to lay down new bony matrix all through this tissue. So as long as these chondrocytes are dividing, then we're going to see that the diaphysis is going to get longer and longer and longer and longer, and the epiphysis just gets pushed up further and further and further and further. And that happens at both the proximal epiphysis, and then if you flip this, it would also happen at the distal epiphysis. So this is how our diaphysis ends up getting longer. We get increase in length of the diaphysis from both ends through this epiphyseal growth. Now, once the proliferating zone of cartilage slows down cell division, the ossification zone never slows down. So ossification is going to keep happening, and eventually it will catch up to the proliferating cartilage and even the resting cartilage. And when this fully ossifies up here, that's what's going to lead to our epiphyseal line of compact bone. And that's when we've stopped growing. So your growth plates are these epiphyseal plates of cartilage. In this X-ray image, it looks like these bones are broken, but no, they're not. This is cartilage. Cartilage does not show up on an X-ray image because it is not dense. Remember, cartilage is mostly made of water, and that's not going to show up on an X-ray. So this is actually, you're looking at the epiphyseal growth plate of a child here and here and here. And we can measure these growth plates and often make predictions about how much more a child is predicted to grow from measurements of their growth plates. Eventually, right, as I mentioned, the growth plates will completely ossify, and that's usually in early adolescence when that happens. That's when we've reached our maximum height. There's lots of things that are going to influence the cell division, right? Basically, how fast these cells are dividing is really going to influence how long our diaphysis of our long bones get. Growth hormone that's secreted from your pituitary gland is going to influence cell division of those chondrocytes. Sex hormones are also going to influence cell division of those chondrocytes. There's a surge of sex hormones that happens in the body of teenagers typically at the onset of puberty, and that usually leads to a spike. We call those growth spurts. That's typically the last major growth spurt that we'll experience in our lives, and it's kind of the beginning of the end. The surge of testosterone is going to lead to rapid growth, rapid cell division here, but immediately following, we start to see a slowdown in the proliferation of these chondrocytes. And as the cell division slows down, the ossification center catches up, and eventually we end up with these epiphyseal lines in our long bones.